The aim of this experiment is to assess the repellent activity against a selection of micropredator species, such as sea urchins, mollusks, and others. Artificial food is prepared on window screen molds and allows us to quantify the amount of food eaten by the micropredators in the controls and the treatments. By using an analytical balance, we weigh known amounts of agar and freeze-dried pulverized algae. Known amounts of natural products or extracts to be tested are added to a mixture of freeze-dried algae. This mixture will also be mixed with distilled water and agar. For the control experiment, only the solvent is used, diethyl ether, which is poured over the pulverized algae. As for the treatments, the extract to be tested is previously dissolved in the solvent and then it is placed on the algae. In both cases, we wait until the solvent is evaporated under the hood. While the solvents are being evaporated, we start with the preparation of artificial food molds. These molds are made of metacrillate sheet with marks that indicate the place where the food strips must be cut. Over the metacrillate sheet there is a wax paper and there is a window screen on top of it. On the window screen, another mold of metacrillate is placed where the artificial food will be poured over. Every part of the metacrillate mold is divided into 3 regions to be used in the 3 experiments. One side of the metacrillate mold will be used for the controlled experiments and the other for the treatments. A known volume of distilled water is added to the beakers containing agar and algae with a graduated pipette. The mixture of water with algae and water with agar is gently stirred in each of the beakers. In the microwave, the beaker with agar is heated to the boiling point. Once the agar is heated, the mixture with the water with algae is added in the beaker and the final mixture is stirred until complete homogenization. Only a few seconds pass between this step and the next one in order to avoid gelification of the agar outside the molds. The mixture of artificial food is poured in the spaces of the mold. The food is uniformly distributed in the mold. This is done with the artificial food for the control as well as with the food for the treatments. After some minutes, the agar jellifies and molds are removed. 2 strips of food are then obtained, one for the control and one for the treatment, both of them attached to the window screen. "15 strips of artificial food are cut to be used in the 15 replicates for each experiment. The simultaneous occurrence of food control and treatment allows us to perform an experiment of feeding preferences. Experiments are carried out in the wet lab. Each experiment consists of 15 replicates. Each replicate consists of a bucket with seawater with a sea urchin inside. Artificial food strips are offered to sea urchins. Sea urchins are placed with their mouth on the strip in between the 2 options of artificial food. The experiment ends and is evaluated once half of one of the 2 types of food is consumed or after 48 hours. Food strips are removed from the buckets and they are observed under the binocular microscope in order to count the number of eaten squares from the window screen. With this data, after making the corresponding statistical analysis, we will be able to assess the occurrence of feeding preferences in the experiments.